Well, hello and welcome to the QEM Heroes webinar series. I'm Christine Wood and I serve as the Assistant Program Director for the Heroes Fellows Program at QEM. And for more than 30 years, QEM has been a national catalyst for STEM education transformation and research capacity building. Currently, the QEM network serves nearly 900 colleges and universities and reaches more than 8,000 individuals nationwide. And this webinar is part of the HEROES initiative. The HEROES program brings together researchers with experience in the federal government to lend their expertise and support to the STEM research and education communities. Our fellows are here to provide tools and tips for navigating the funding landscape, developing STEM programs and partnerships, and improving talent retention in in-demand fields. We hope this series promotes engagement with our HEROES fellows and provides valuable resources to our participants. The webinar recordings and slides will be available in the QEM Resource Hub. By attending this webinar, you will be added to the QEM network and granted access to the Resource Hub where you can view all of our past webinars. And I'm so happy to introduce our Heroes Fellow today. Dr. Yulia Goreva is a geochemist by training with a PhD from Caltech, specializing in earth and planetary sciences. She has previously served as a scientist at the Smithsonian National Museum of Natural History and as a scientist and technologist at NASA's Jet Propulsion Laboratory. So without further ado, I will turn the floor over to Dr. Goreva for our presentation today.
Hello, everybody. Thank you so much for being here. So today I will continue the series that I started a few months ago on on-ramps for mostly PUI institutions or non-R1 institutions in the fields that may seem intimidating. So we're going to talk about astrobiology. So why this webinar exists? So first, the science is wild right now, and hopefully I'll show you what that means. Second, this field needs you and your students. Most of astrobiology is not a flagship hardware. It's a data analysis, modeling, fieldwork, sample analysis, training students, and so forth. So, as I mentioned, this is part of the series, but I want to draw your attention that next week there will be a webinar specifically about engaging undergraduate students. So we will discuss we will discuss the strategies for involving students in the research and of course, finding funding strategies and grant opportunities for each. So the field that I'm going to talk about today is astrobiology, and I'm going to go through four sort of sections in the next half an hour, 40 minutes. And I know most of you came here for the second two, where we fit and where the funding is. But I won't rush sections 1 and 2. I will keep them tight, but I want to set up a framework of why and how you can engage students and actually start a research group or research program, starting only with one student. If you have any questions, please, I think, Christian, we are accepting questions in the chat, right?
We are accepting questions in the Q&A box,
And the
which will
Q&A.
be enabled.
Okay, thank you. All right, so astrobiology. It's rooted in three questions. How did life begin? Are we alone? And what is the future of life? Astrobiology is the field that, or the science that takes all these three questions very seriously. It translates them into experiments, missions, and observations. Why I decided to bring this particular field to your attention is, well, because of these three things, and they're all very recent and all transformative. James Webb Telescope First time we have spectra of habitable zone Earth-sized exoplanets. This is huge. It's transformative. Ocean worlds. Europa Clipper is the highest priority NASA flagship right now in decades for astrobiology. Dragonfly, I'm going to talk a little bit about this later, will fly to Titan. JUICE mission is European, but contributes massively to the same question of the ocean, exploring ocean worlds and the other planets. AI, artificial intelligence, machine learning. This is genuinely a paradigm shift. NASA's DARE strategy explicitly calls out AI/ML as a transformative capability. So that might be an on-ramp for any computer science faculty or computer science department. Astrology is a team sport. So I'm not going to read this list, but you can just look at it. And the cumulative effect of that is that you have to realize that you might be fitting somewhere or maybe in several of those categories. If you teach in any of those fields, and most of you probably teach at several, there's a place in astrobiology for your students. Some key vocabulary. I'm sure that you know the difference, but I'm just gonna highlight them because I'm probably gonna throw them throughout the presentation and don't want you to sort of get lost. So habitability is a condition that could support life, and it's not the same as actual life. Please do not conflate those. Biosignature, a chemical, physical, or pattern signature attributable to life. Again, it's not a life. It's a signature that can be attributed to life. There's another term that technosignature. This is completely different. Also, do not mix with that. The technosignatures are signatures of advanced technological advanced technology. And it can be radio waves, it can be some laser shootings, and so forth. Abiogenesis, the origin of life from non-living chemistry. Basically, having a building blocks that can build life. Agnostic biosignatures. We are, by definition, Earth-centric. So looking for life, we tend to look for life as we know it. Agnostic biosignature is something that love-like patterns that do not assume Earth biochemistry. Extremophiles, organisms that surviving or thriving in conditions that would kill most of life on Earth. Planetary protection is a program or way to prevent biological cross-contamination between worlds. And standards of evidence is basically agreed upon criteria for what counts as actually detection of life. I'll use those terms throughout, so just wanted to outline them. All right, so Mars. Perseverance cached just over 30 sample tubes on Mars in Jezero crater, including samples from Chiava Falls. And I included two images here. Upper one is a Chiava Fall. And this sample, it was scored right where those leopard spots are in the middle of the image. If you can see the whitish, kind of roundish spots with the black rims. These kind of features, if on Earth, that would be they would be very telling about fossilized former life. We don't know if it's the case for Martian rock, of course, but this rocks also has organics, detected organics by in situ instruments. It has hematite, it has phosphate. All of those are little tell signs that this rock would be so good to have back in the lab on Earth, where bigger and stronger and more precise and more sensitive instruments are available, much more precise than what we actually sent to another planet. So below is a tube sample, actually sample tube containing a core, drilled core of the Martian surface. And these tubes right now on Mars waiting. The MSR program is on hold, but hopefully the samples will come back. And this matters because the Mars sample analysis, when it returns, will be distributed across hundreds of labs, and your students might be the ones who actually would be analyzing the Martian rocks. Rosalind Franklin, formerly ExoMars, now named in honor of DNA Frontier. And if you don't know, Rosalind Franklin played an integral role in the discovery of the structure of deoxyribonucleic, acid. It was her X-ray images that allowed James Watson and Francis Crick, who actually got a Nobel Prize, to decipher its double helix shape. Unfortunately, Franklin's early death from ovarian cancer meant she never received the recognition. Recognition was given to her male peers, but this is her recognition. The ESA rover, deep drilling rover, is named after her. So Rosalind Franklin will drill 2 meters into subsurface of Mars. So compared to Perseverance, Perseverance's cores are basically depth of those sample tubes. So it's about like up to 10 centimeters, 6 to 10 centimeters deep. This rover will drill 2 meters. Why is it important? Because this is way deeper than radiation effect and sterilization effect of the cosmic radiation on the very surface of Mars. It was scrapped originally the launch was scrapped in 2022, mostly of geopolitics. It was formerly a mission involving Russia. So they're no longer involved in the mission. And now the rover is planned sometime this decade, like in the late, probably next decade. So the key distinguish is that this rover drills deeper than Perseverance. And where the cosmic radiation does not degrade organic molecules. So if there are preserved biosignatures in Mars subsurface, this would be our best shot. Europa Clipper, currently en route to Jupiter. It launched in 2024, arrives to Jupiter 2030. It will pass Europa 50 or plan to pass about 50 times. It's not orbiting Europa, it's flying past it from Jupiter orbit, sort of like a slingshot. This is, I want to clarify something because I've seen in the media that was misreported. This is not a life detection mission. It's a habitability mission. So it's asking, could the subsurface ocean support life? Not is there life over there? And this distinction matters because the next NASA mission, a Europa lander actually, and it's currently in concept, would be a life detection mission. The other ocean worlds, Enceladus. Cassini already did the impossible, flew through the plumes, and put a picture over there of ocean plumes, and tasted the ocean. It found organics, including methane, that could be biological or could be just hydrothermal. Found salts, so we know it's a salty ocean. It found silica grains, suggesting active hydrothermal vents, kind of the environment where life may have started on Earth. And we do know that there is life in hydrothermal vents on Earth. And the Titan Saturn's largest moon. Well, this is the strangest body in the solar system, in my opinion. Most alien place, liquid methane lakes. The atmosphere is nitrogen like Earth's, but chemistry is utterly different. I included here a picture of a dragonfly mission. It will be a literal rotorcraft on Titan hopping between sites and measuring prebiotic chemistry. This technology is possible because of Titan. It has low gravity and very dense atmosphere. So you have this kind of like a hopping dragonfly, literally. A very good name for that. It launches into our plan to launch in 2028 to explore Titan. Next frontier, actually accessing an ocean. Clipper, Dragonfly, JUICE, European JUICE will all return remote sensing data. So the question after that, how do we actually get into the ocean? And there are three currently concept families. Europa Lander, it's a surface mission, drill about 10 centimeters, around live detection chemistry. Enceladus Orbilander would thought to sample the plumes and the south polar terrain. And the cryobot, kind of like audacious one, I love this one. It's to melt. It's basically like a cylinder to melt through a kilometer of ice to reach liquid water. Currently being developed at JPL and elsewhere. So the point what I'm trying to make is that undergraduates, your undergraduates today will be postdocs and PIs flying, possibly flying these missions. These are all concept studies right now, but the pipeline that we established, the student pipeline, matters for science. That's what matters for science. Exoplanets. As I mentioned, James Webb Telescope changed everything. When the planet passes in front of its star, a starlight passes through the planet's atmosphere. Different gases absorb at different wavelengths. So the missing wavelengths in the spectrum tell you what gases they are. And I included here transmission spectrum of WASP 39b, I believe, captured by Webb's near-infrared spectrograph a couple of years ago. It reveals that there's a rich soup of molecules in a planet's atmosphere, including first detection of the light-produced molecule, sulfur dioxide. So the blue line, blue line is a best fit model, but the scatter points is actually real spectra that James Webb obtained. And the highlighted peaks is actually the molecules that we now detect in the atmosphere of other planets around other solar systems. So what's next? Habitable World Observatory. This is NASA's next astrophysics flagship. It is the top recommendation of the 2020 Astrophysics Decadal Survey. Currently, it's in early formulation. STARS, which is Science Technology Architecture Review Team, is doing the planning. Launch realistically in the 2040s. Same point. Undergraduates today will be working scientists, hopefully established scientists already when this launches. So recruit them now. Origins of life. The ingredients are everywhere. Three data points. Bennu samples, which are OSIRIS-REx samples that OSIRIS-REx returned in 2023, contain nucleobases, and the building blocks of DNA and RNA, plus amino acids. What's important is also that the sample is from primitive carbonaceous asteroid, the fairly typical asteroid for asteroid belt, telling us that the chemistry, this prebiotic chemistry, is widespread. Paolo Hopkinson, a very recent study, Aarhus University in Denmark lab shows that peptides, which are precursors to proteins, form on the surface of interstellar dust grains before stars and planets even form. And of course, one of my favorites, Murchison meteorite. This is the gold standard meteorite for prebiotic chemistry. Decades of work showing evidence of various amino acids, sugars, nucleobases, all are presumed to have abiotic origin, but this is all life precursors. So starting chemistry for life is delivered to rocky planet pretty much all the time, to ours and to Mars and to others. Earth-based, so we're gonna move a little bit closer home now. Earth-based astrobiology, extremophiles. You can do the legitimate astrobiology in your own region without the spacecraft. Most extremophile work uses standard departmental equipment. RNA sequencing, sometimes it can be outsourced cheaply, but the sequencing setup is not very complex. It's standard microscopy, basic biochemistry, and field sampling. This is probably the most expensive piece, but I'm going to talk a little bit about like Lewis and Clark Fund, and that might be covering that in a funding session later on. So if you teach in a region with hot springs, salt flats, caves, glaciers, deep mines, or just unusual soil microbiomes, you can publish in astrobiology, in the Astrobiology journal. AI, artificial intelligence, machine learning, and agnostic biosignature detection. So traditional biosignature search is look for things like Earth life. I talked about this in the beginning. Agnostic biosignature search is look for things that look organized in ways chemistry alone wouldn't predict. So processing a big amount of data and finding patterns is what AI is best for. Why this matters? This work is mostly data work, not lab work. So computer scientist or statistics faculty member can be very productive on day one and teach their students to do that. Open NASA source is for that is LDKB. It's a live detection knowledge base. And it is AI is one of decadal astrobiology research and exploration strategies, or theirs. Planetary protection. This is a cross-cutting frontier. It's rooted in two questions, that is forward contamination and backward contamination. The idea is to protect worlds from cross-contamination. Forward contamination is prevent Earth microbes from contaminating Mars, Europa, and Enceladus before we look for native life. And it can be to protect the worlds, and it can be protecting science. For example, if we return samples from Mars, we have to be absolutely sure that those sample tubes that I showed you did not come to Mars with Earth microbes. Because then those would be confused. The scientists wouldn't know whether they came actually from Mars or it's hitchhikers that came from Earth to Mars and then back to Earth. Backward contamination is when we return Mars samples and eventually, of course, Europa and Enceladus samples, we have to assume that they may contain biology and contain them appropriately. And that's the part of backward contamination policy for planetary protection. Again, you don't need clean room to study. To study how some bacteria like Bacillus ceblitis spores survive simulated Mars conditions. You can do it in departmental incubator. You need controlled environment, and a student or two. So where the field is heading? NASA's DARE strategy, which is a decadal astrobiology research and exploration strategy, being finalized through this year, 2026, first time in a decade. This is a document that will shape NASA astrobiology funding for the next decade. It's being built through open community input. So Task Force 2 webinars around February through March. So this is all like very, very new. And I wanted to highlight 9 focus areas here. I'm not going to read them, but I want you to look at them and see that it goes from RNA science or biosignature to system level area like star-planet interaction to astrobiology and society and science communication and education. So these 9 areas span everything from RNA chemistry to science communication. And the last 2 are on ramps for humanities and social science faculties. So again, you don't have to be a planetary scientist or astrobiologist or even a biologist to actually contribute a very meaningful research to astrobiology. So this 2 points, 8 and 9, are actually very unusual for a science strategy document, and I thought it's worth highlighting.
Dr. Goreva, we have a question that is relevant to this slide. What is omics?
Where is it? Sorry, I have my blocks, slide blocks blocked. Oh, uh, hold on a second. Oh, it escapes me right now. I'll answer it. Yeah, I'll answer it a little bit later. I'll formulate it and answer it after this webinar. Sorry for a hiccup. Kind of got lost my train of thought. All right, the myth and reality. That's basically how I built this. Wanted to dismantle the myth that astrobiology is for big missions and R1 institutions. And the reality that most James Webb Exoplanet papers are written using publicly archived data. Mars rover data is on planetary data system, PDS, free for everyone, and also most data, a lot of papers are not directly tied to the mission. The most planetary protection papers use standard microbiology equipment. So competitive disadvantage of being at PUI is real. I don't want to be all rosy eyeglasses, but it's not the equipment. It's time, network, and confidence. So I'm going to try to address these 3 in the next few minutes that we left. I'm still thinking about more omics. I don't know why it escapes me. Sorry about this. All right, so 6 on-ramps that don't require lab. I'm not going to go through all of them, but just highlight a couple. Archival data analysis. This is probably the easiest entry in the field. MAST here would have a Webb and Hubble telescope data. PDS for planetary missions, and of course, NASA Exoplanet Archive. It's all publicly available data. Modeling for computer science, Rogue 3D is NASA's open source 3D climate model. There's another one with a funky name for the atmosphere retriever. It calls Petit Trans. It's a long acronym. NASA likes acronyms. And that's for atmosphere retriever. Again, data is all free, but it needs to be processed. And probably my favorite is extremophile fieldwork. So if you're a faculty member in Utah, it's Salt Great Lakes. New Mexico has caves. Alaska has glaciers. Most regions have something. So you can do field work, you can do, or you can start astrobiology research. Very easy, uh, single course. Well, it's not very easy, but it's a single course. It's a really lowest cost, highest impact way to start. Astrobiology slots into existing departments, so you really don't need to have a major in your institutions to start a program and actually involve students. So I put a few examples that might work and resources that you can use to actually start that. And it can be an upper division astrobiology course uh, for the next academic year, no grant required. But even if you recruit one STEM student in the astrobiology field, this is the beginning of a research program. A senior research seminar centered on archival data analysis projects, also really a good way to involve students and set them up for master's or PhD that they may pursue in astrobiology. Philosophy of Science seminar on biosignatures, evidence standards, life detection ethics, communication, and we've seen many hiccups in media about life detected or search for life and on Mars and so forth. These are all humanities or building humanities students to support astrobiology. And free resources, astrobiology learning progressions. Use it. It's free. It's built by NASA. You can use it for any class that you can build. Really nice documentary, Our Alien Earth, also by NASA. Uh, just watch it with students or point them that direction, and a couple of textbooks that kind of useful to get students started. Big one. I've talked about this before. Partner with an R1 or NASA center. These partnerships exist because somebody just made a phone call. And I put two concrete examples here, Cal State LA and JPL. This is a decade-long collaboration. The mostly teaching-focused institution sends undergraduate interns routinely to JPL as a part of Origins and Habitability Lab initiative. Multiple first author student publications, and it's funded under NASA SMD Bridge program. Aspire, another one, Skyline College, San Jose State, NASA Ames, Community College, Regional State University, and a NASA Center. Undergraduates routinely contribute to Origins of Life and exobiology research, also SMT funded. Both these are like big established collaborations, and both started with a single email or phone call, I think. Sure, about UpSciCon. UpSciCon is a biennial, so unfortunately, it's too late for this year to submit a poster or abstract. It's this May soon in Madison, Wisconsin. The next Astrobiology Science Conference is in 2 years. It's not announced where yet. It's going to be in 2028. Just practical tips. Posters are usually accepted up to 3 months before the conference. So the deadline is in late winter. There are travel grants. There are multiple programs like NASA, the conference itself, AIS sometimes, multiple ways to get travel grants, especially for students. Undergraduate posters are welcome, and not just welcome, the reviewers are especially kind to them, so do that. And I have to mention UpGrantCon. This is a spin-off that is run by graduate students. It's a student-run version of UpSciCon. It is yearly. It happens annually, and it is incredible for students starting in the field and actually building a network with their peers. So I wanted to bring attention to that. Getting students into pipelines. So these programs below send your students to NASA centers and major research institutes for the summer. Deadlines mostly January, February for following summer. So again, we are a little bit late for this year's cycle, but you should be getting recommendations letters now for next summer. So this right now is a time to work for next year cycle to get the grad students for undergraduate students for summer internships. Space grant. Every state has one. Some are very PUI friendly. They fund both faculty mini grants and undergraduate research stipends. So look up your state space grant office name and have it ready. I can guarantee that if that one of your current undergraduates is qualified for one of those programs, but you have to sort of go through these loops, get recommendations, and apply. The funding landscape, probably what most of you are most interested in. And I split it into three categories, the low barrier, mid, and high barrier funding. So this is a lower barrier. So start here if you are starting a research program. Small dollar amounts, simple proposals, friendly reviews, often discipline agnostic. This is the most under-promoted tier in academic culture. Most faculty thinks real grants mean 300K, NSF, NIH, NASA, but miss the small dollar high yield programs entirely. You have to start here if you're starting a program. Lewis and Clark, NASA funds, they're small, they're 5K. The applications are essentially two-page proposals, but it can fund a graduate student summer field season. And it establishes or starts establishing your credibility in the field, you as a faculty, and of course the student who actually doing the work. So wins at this tier build a track record for what you're actually going to do next. Medium barrier is a working horse. Real research grants, 100 to 500K typical, writable by an individual faculty PI. If you want to take anything from the like really on-ramp funding landscape is to take home previous slide and NASA SMD Bridge program. NASA SMD Bridge is the flagship NASA program for partnerships between PUIs, MSIs, HBCUs, and NASA centers. Track records includes the Aspire program, Cal State LA that with JPL collaboration that I mentioned earlier. So start there. And of course, there are other programs. NSF RUI, it's a destination, not a separate program. You apply to the regular NSF program with an RUI designation, and reviewers will know your context. Our word sizes are usually smaller because budget PUIs are smaller, but the success rate is fairly high. And of course, Rosa's exobiology, the standard NASA astrobiology competitive, would not recommend starting there, but this is something to shoot for. Last line, NASA SBIR, STTR. We had a wonderful presentation yesterday that that went specifically into this category of funding. So I'm not going to cover that, but if you're interested, please look at the QEM site for this presentation. It actually outlines the strategies to obtain that funding. And I summarized it here, but again, I'm not going to double myself, but you'll have it for your reference. High barrier, this is the long game. Large multi-year team grants, realistic timeline is 5 to 10 years minimum into your astrobiology engagement, often as co-PIs or an R1 led on an R1 led team. We'll keep it brief. Most of you won't be writing these proposals right now, but you should know that they exist. Just know that they exist as a destination. Private foundations, much less paperwork than federal grant application, often willing to fund unconventional or risky work. Key distinctions from federal funding, the Heising-Simons Foundation, that's probably explicitly more risk tolerant than NASA. So if you have crazy ideas, apply there. Templeton is unusual in that it funds humanities and plus science crossover work, so including in astrobiology. Look up there if you are in the humanities. Cottrell Scholar Award is the gold standard for PUI faculty in chemistry and physics, and astrobiology overlaps both. So I wanted to bring it up, but don't choose between federal and foundation funding. Run both pipelines in parallel. And a brief strategy for first-time PIs. First, call the program officer before you submit. They want to talk to you. They're real people. They have phones and emails, and they actually pick up. Most academics don't realize how much of getting a grant is a conversation with the program officer before you write. And the reason is because you want to make sure the proposal that you submit actually aligned with the program goals and mission. Track record matters. Reviewers are reading hundreds of proposals. They look for signals. A previous grant, even a 5K one, signals this person can win money and execute. Next one, I covered that co-PI with R1, not because you cannot do it alone, but because the network signal helps reviewers. Write the broader impact section like you mean it. PUIs have natural advantage here. You teach undergraduates, you're often in underserved region. You serve students who are first generation. Write this. Reviewers, this is not a disadvantage. This is not an excuse. This is truth, and this is your advantage. Reviewers rewarded. Funding, target 2 tiers up, not 5. This is a strategic punchline for us, for all of us. Many faculty try to write a large proposal as their first NASA submission because they have this grand idea. Very often they lose. Most likely they will lose. Do the steady climb instead. Skipping ranks is how first-time good proposals sometimes lose to mediocre ones from established teams. Right, and resources to take with you, just few links, hubs and strategy, and astrobiology site. Excuse me, there's strategy and task force webinars and recording. community news aggregator for at astrobiology.com, and the funding links. But I think previous slides are covered in more detail, so you should be able to find information there. Main conference is UpsideCon, but of course there are other that are planetary science or even AGU, American Geophysical Union, that overlap with astrobiology. So AGU is San Francisco every year. Lunar and Planetary Science Conference, Houston every year. Meteoritical Society, if you do astrobiology related to meteorites, MetSoc meeting every single year in the summer. So get your students to submit a poster to one of those. This is how you start them. And look up that UpGradCon also for students. This is wonderful, wonderful way to build up a network, not by students or not only by students approaching a big name faculty member, but actually building a community of astrobiology students from the students up. So it's a networking, both science and networking event. And this is it from me. I'm gonna leave you with codes for to our QM events page and for upcoming events. Uh, and again, I'll plug a little bit for our specifically undergraduate involvement of undergraduate webinar on May 12th next week. Here's webpage and a feedback form if you choose to leave a feedback. Thank you so much.
Thank you so much, Dr. Goreva. We do have time for questions. You can ask a question by inputting one into the Q&A box or by raising your hand to come off mute. And I wonder, Dr. Goreva, if you want to approach the omics question, or we'll have to get back to that at another time.
What questions do you have also?
Well, Greg Davis asked, what is omics?
Well, I was trying to formulate it, but basically it's a field of study in biological sciences that focuses on this very large scale quantification. So it is a pattern recognition, but it's a really large scale quantification of biological or chemical molecules. And I do not remember, I honestly do not remember where the name comes from, but this is the concept, Greg. Thank you for your question. And sorry for kind of getting hicked up during the talk. But it is a large scale pattern recognition method or field of study in biological sciences.
And from Dr. David Richards, per Wikipedia, the branches of science known informally as omics are various disciplines in biology whose names end in the suffix omics, such as
There
genomics,
you go.
proteomics, metabolomics, metagenomics, genomics, and transcriptomics.
That is actually funny. Yeah, I honestly didn't know.
Dr. Goreva, I have been thinking throughout your presentation about if I were a faculty member at a predominantly undergraduate institution, what would be the argument for attracting an undergraduate student to this field? Some people might think of it as something that's philosophically out there. What is the benefit for us on Earth to be studying astrobiology and getting involved in it?
Well, this is a philosophical question, right? We as a humans are designed to explore new worlds. And I'm going to talk a little bit careful because I don't want to speak for all scientists, and I can express my own view. But there are some that would think that or do believe that exploring new worlds, just like Columbus came to America and going with humans to a different planet is the part of how we humans evolve and think. So astrobiology fits in in that sort of in that niche, uh, uh, the search for life, whether it exists elsewhere and how it started on Earth, are fundamental questions to understand, because that actually may um, lead us to understanding where are we going as species. Right. So if you know where we start, and if we as a species can start somewhere, or other species can start somewhere, or finish our existence on one planet and continue to on another. So it is philosophical. It's a fundamental and philosophical questions, but are rooted in a real science that can be done.
We have a little bit of time for additional questions. Please don't be shy. Use the Q&A box or raise your hand to come off mute. And we have Gregory Davis. Greg, you're able to come off mute?
Yes. So I keep thinking about this sample collection on Mars. It sounds really cool. And I wonder what the retrieval mission would look like. And in particular, I wonder why it is that we felt like it was a good idea to create the samples now. What kind of mission would we have that could collect the samples off the surface but couldn't collect the samples individually? Uh, I was just I can't picture that, and I would like to.
What do you mean, Greg, by individually?
Well, so we're going to send a mission to Mars that has the capability of grabbing these cylinders and flying them back to Earth, we think, right?
That's
So,
right. So that was original concept of MSR, the Mars Sample Return. And yes, it was actually evolving program, and that's why it ran over budget and was put on pause on NASA. And that's right now, solicits a private corporations or private companies to design the mission for return. But yeah, it's got to be a mission that uh, is this and I'm going to start answering, Greg, and you correct me if I'm answering not your question, okay?
okay,
So so uh, to send a mission which is both collection and return is incredibly difficult, because there are just way too high of a risk of a failure, uh, or too many failure points. So Perseverance, which is a Mars 2020 mission, is designed to collect, and it landed its first time it drills Martian surface. It collects samples, or collected samples, in ultra pristine, ultra clean titanium tubes that are hermetically sealed. I was actually involved in designing those tubes and verifying their cleanliness and so forth. So it deposited about 10 samples on the surface of Mars, uh, and has about 23 right now on board. So it's still exploring Mars, but it has a number of samples on board. So for the retrieval mission, there are two options right now. You can sort of like use it as a like a pooper scooper. I came to the first depot location and collect the samples that are carefully documented and sitting on the surface. I showed a picture of a sample tube actually just dropped. Um, Here it is below. This is how it is. So all you need is this kind of a hand, a robotic hand, to pick them up. And then the from technology side, the most difficult part for that mission is to take off Mars, because we've never done that before. We need to
right.
launch. It's not just collecting samples. We need to launch from Mars and then come back to Earth. So that's one of the concepts. Another concept is a little bit more complicated, that that requires interaction with Perseverance rover, and that's why some of the samples are there, are still on board. But this is also a way to shield the samples from excessive heat on the surface, for example, that may in part deteriorate science. In the second concept, a smaller rover or lander either comes to Perseverance, or Perseverance, if it's alive, drives to that rover, and then a hand or handling robotic hand retrieves the samples from Perseverance, puts on board, and then again launches from Mars. So there were several concepts that were proposed and worked on as a part of MSR, and they were upscoped, descoped, but eventually the program was overblown financially, and that's why it's put on pause, because it started, as much as I would hate for the samples not to be returned, I also appreciate the fact that NASA cannot put a majority of its budget into one program only, right? It would have to be done at the expense of other programs. So it's a risk-benefit analysis. But samples are there, and I hope, I don't know, it can be Musk, it can be some other program that will actually design a mission that can bring them to the lab. Did I answer your question, Greg, or I answered my own
Yes,
question?
you said an awful lot, and a lot of it is interesting. It just seemed to me that if I put together the capability to get a rocket onto Mars that can launch off and come back to Earth carrying samples, that I could also put into that mission a rover, and then I would be able to choose my own samples instead of getting the ones that somebody else collected years ago. Um,
Well, somebody
but
else corrected and choose your own sample, Greg. The samples that were chosen, it's it's a community effort. So it's hundreds, the science team for Perseverance is hundreds of international scientists who, before collecting a sample, investigated very, very carefully. To have a mission that has both landing, collecting, and taking off is an effort that just cannot achieve that. Because before collecting, before recording, like this image that we have on top, uh, before choosing to collect a sample, before actually drilling, the portion of the rock is abraded by the abrading tool. It's kind of like a sandpaper that clears the surface of the surface, weathering, and just sort of gets a few millimeters down. And then very good high precision instruments on at the end of the robotic arm of Perseverance go very close to that abraded patch and do chemical analysis and spectral analysis and all kinds of analysis on that patch. Then hundreds of scientists digest or process that data and then make a decision, weighted decision, because there is a limited number of tubes on board, weighted decision whether this is the sample that is worth bringing to Earth. So collect my own samples and bring them to Earth would work, but I don't think it would be as thoughtfully chosen as Perseverance does right now.
okay, thank you.
Wonderful. Thank you, Dr. Goreva. This was so interesting and lots of thoughts for getting undergraduates involved in this research, but lots of insight into the value of astrobiology and what it's all about. So I want to thank everyone for coming today. We hope to see you at a future webinar, and you are free to go to our resource hub and access all of the other webinars we have up there too for your edification and learning and resource sharing. Thank you so much.